Even in a non-pandemic year, respiratory infections are one of the biggest killers worldwide. These diseases impose an immense burden on health, particularly in the developing world. But how those pathogens are spread when we cough, talk, and even breathe is barely understood. That's because current systems, such as the rotating drum, have their limitations. With this novel system, Celebs, we can mitigate those limitations, better understanding disease transmission, helping to inform public health, and to help save lives. This project looks at a surrogate for COVID-19, mouse hepatitis virus, and how that survives the first 5 minutes of aerosolization, what improves that infectivity of that virus, and what reduces the infectivity of that virus. Celebs is a single particle levitation system. We begin by generating a reproducible bioaerosol, which is caught between two electrodes in an electrodynamic trap. After the intended incubation period, could be 5 seconds, could be 5 minutes, we collect onto a substrate by releasing the electrodes. So with this lateral view, we can see we've got 5 droplets caught in the trap, and that's how we enumerate. And with this planar view, we can make sure of efficient capture, make sure no droplets are flying away. This is a virus, so we need an infectivity assay. So we incubate the collection substrate with mouse cells. So C is a negative infection and D is a positive infection. And this way we can calculate the virus per droplet, but also calculate the viral infectivity. So in the early days of the pandemic, papers were reporting that COVID can survive in the air for hours. But what we actually find is that even in the first 30 seconds, there's dramatic loss of infectivity, particularly at lower RH. We do see some loss of infectivity at higher RH at 90% after about 5 minutes. To make my bioaerosols more representative of true life, I introduced clinically relevant concentrations of mucin. And as you can see, at 30 seconds, there's increased concentrations of mucin, increase the infectivity. This is all kind of pointing towards phase change driving the loss of infectivity. So when the droplet is drying and efflorescing, that is causing the virus to die. But with this loss of infectivity at higher RH, that's pointing towards other mechanisms which we yet to understand. So this research demonstrates the time resolution of the Celebs study system, also demonstrates the importance of measuring single levitated particles in the early lifespan of the bioaerosol, and the relevance of incorporating biological factors, in this example, mucus. So hopefully I've made the case for Celebs. I'd like to thank everyone in the aerobiology group that I work with, and thanks for listening.